So the process by which the information in the RNA is converted into a protein is called translation. And the whole process of translation, it goes about in 3 different steps. The first is initiation. The second is elongation. And the third is termination. Now, these steps are similar to that of the transcription, because transcription also occurs by involving 3 different steps. Now, in this video, we are going to specifically talk about the initiation process in eukaryotes. So, before we start off with the initiation process, the players that are involved in the process are mainly the ribosomes, some factors which are called initiation factors and are written as EIF, and they have different numbers. So the E is for eukaryotic and IF is for initiation factors. And finally, the tRNA, which brings in the different amino acids. Now, there are 2 independent events that occur during the initiation process. The first independent event that occurs is the preparation of the small subunit of the ribosome. Let's say this is the small subunit of the ribosome. We know that the eukaryotic ribosome is 80S and the larger subunit is 60S and the smaller subunit is 40S. So preparation of the smaller subunit, that is the 40S, is the first independent event that occurs. What happens is that there are 4 different initiation factors. 4 different initiation factors. The first factor is EIF1. The second initiation factor is EIF5. The third initiation factor is EIF1A, and the fourth initiation factor is EIF3. These 4 initiation factors, they work in tandem and binds to the smaller subunit of the ribosome, thereby preventing the smaller subunit to associate with the larger subunit. So that is the main goal. These 4 different initiation factors, they bind to the smaller subunit and prevent it from binding to the larger subunit. Another initiation factor, which is called EIF2, it brings in the tRNA bound to the initiator amino acid, that is methionine. So in case of eukaryotes, we don't have formyl methionine or FMET, we only have initiator methionine. So it is written as MET-I. Now, this EIF2 can bind to initiator tRNA or MET-tRNA-I only when it is in a GTP bound state. This is very important. The EIF2, it can only bind to the initiator tRNA and bring it to the smaller subunit when it is in its GTP bound form. Now, this brings the initiator tRNA and places it in the region of the 40S, which is supposedly to be the future P site. So the initiator tRNA, it is bound somewhere over here. And one more thing, when this EIF2 is in GTP bound form and it is bound to the initiated tRNA, this whole complex is referred to as the ternary complex. This brings in the initiator tRNA or the met tRNA I onto the P site of the 40S. Now this whole complex with the initiation factors, the initiator tRNA, EIF2, all of that, it makes the 43S initiation complex. So now the first step is achieved, the prepping of the smaller subunit of the ribosome. Next, what happens, in a separate series of reactions, the mRNA that is needed to be translated, that is prepared and brought to the smaller subunit. So we have the mRNA. You have the mRNA over here, and since we know that the eukaryotic mRNA has this 5' cap, it has this 5' cap. Now this cap of the mRNA, this is recognized by another initiation factor which is called EIF4E. Now this recognizes the cap and binds to it. So this is bound to the cap. Now once this EIF4E is bound to the cap, it recruits another initiation factor which is called EIF4G. So it is recruiting another initiation factor which is called EIF4G, which binds to the EIF4E and the mRNA. So EIF4G. Next, this EIF4G, it recruits another initiation factor which is called EIF4A. So this is EIF4A, which binds to EIF4G and the mRNA. Now once these three initiation factors are bound to the mRNA, another factor which is called EIF4B, this stimulates the EIF4A, which was previously bound to the mRNA and EIF4G, to have an helicase activity which gets rid of the secondary structure near the 5' end of the mRNA. So EIF4B, it stimulates EIF4A, that is, it is stimulating this initiation factors, and if there are any secondary structures, let's say you have a secondary structure over here, this secondary structure would be degraded by the helicase activity of EIF4A, which is stimulated by the EIF4B. Now finally, all the EIF4 proteins brings the mRNA, to the smaller subunit, and aligns it with the other initiation factors and the 40S. Now when we have the smaller subunit, this is the P site, this is the A site, and this is the E site, along with the mRNA, and also all the proteins and the tRNA, Now, this complex, that is this one along with this one, when both of them are bound together, it gives rise to the formation of the 48S pre-initiation complex. Now, once this is assembled, the 48S initiation complex, the ribosome, along with all the initiation factors, they move along the mRNA in the 5' to 3' direction and scans for the initiator codon, that is AUG. Now, there is one interesting thing, that this AUG, which codes for methionine, it is recognized by the help of some other nucleotide sequence, that is at the initiator codon you have AUG, of course, but you also have a purine, that is G or A, three nucleotides upstream of AUG, and a guanine just immediately after the AUG. So this sequence, that is the purine at three nucleotides upstream of the AUG and a guanine at a single nucleotide just immediately after the AUG, these nucleotide sequences are recognized by the 48S pre-initiation complex. Now, this sequence is referred to as the Kozak sequence. So, the 48S pre-initiation complex, it recognizes the Kozak sequence and the tRNA, which was present over here, the tRNA, which is present at the supposedly P site of the small subunit of the ribosome, it binds strongly with the AUG. So let's say the AUG is over here. Now the tRNA, or the initiator tRNA, would make hydrogen bonds with the AUG, and this binding, this correct base pairing between the initiator tRNA and the start codon, that is the AUG, that would trigger a conformational change in EIF5. This binding, this triggers conformation change in EIF5. Now, since the conformation of EIF5 is changed, this conformation change stimulates the EIF2. The EIF2, which had brought the initiated tRNA into the complex. So the conformation change of EIF5, it triggers the conformation change of EIF2, which hydrolyzes the GTP that was bound to it, and now is bound to GDP. And as soon as the GTP is hydrolyzed, this EIF2 leaves the complex along with EIF5. The loss of EIF2 allows the binding of a second GTP regulated initiated tRNA binding protein, which is called EIF5B. So now the EIF5 is not there, EIF2 is not there, and another protein that comes in which binds to the complex is EIF5B. Now, as soon as the EIF5B comes in and binds to the complex, all the other proteins that was there, all these proteins, they leave the complex, and only the ribosomal smaller subunit of the ribosome, that is present. Only the smaller subunit of the ribosome that is present. Now, as EIF5B, it triggers the release of all the different initiation factors that were previously bound to the smaller subunit. This sort of helps the larger subunit, that is 60S, to come and join the smaller subunit. Now, binding of the larger subunit, that is 60S, to the smaller subunit, that triggers the release of EIF5B. And now the whole complex is ready for the elongation process of the translation.